So since bio means living system, techno means techniques, and logy means study. So from the word itself, we can conclude that biology is like the study of like the integration of living systems and the components with techniques. So basically, like bringing living systems and techniques, like combining the bio and techno together, like the study of that area. So we learned the definition as biotechnology is the integration of living systems and their components with techniques to produce products and services. Then we learned a simpler definition. It deals with study and development of techniques using living system and its components to produce products and services. Then we learned about some biotechnological applications such as IVF, RNA and DNA vaccines, genetically modified organisms, DNA fingerprinting, gene therapy, gene synthesis, and so on. Then we learned about the principles of biotechnology. So there are two core techniques, that is genetic engineering and bioprocess engineering. So genetic engineering is techniques to modify the chemical composition of genetic material. And so basically like stuffs like gene synthesis and things like that. Basically anything to do with genes, like modifying their genetic material such as DNA and RNA, and introduce it into other organisms to study the phenotype from phenotype. So basically, genetic engineering is like experimenting with genes and observing that what characteristics they provide. Then we learn about bioprocess engineering. This includes the creation and maintenance of microbial, like microbial contamination-free ambience in chemical engineering processes. And the bioprocess engineering is used to regulate the growth of these microbes. It also includes the synthesis of such materials that are used in technology such as IVF and yeah, like that.
Yeah, correct. Thank you so much, Atif. And then I ask you to go back and read about recombinant DNA technology, plasmids, and gene cloning and gene transfer. So I hope you did that. Did you brush up on this, people?
Yes, sir.
Because we will be, it will help you in the discussion in today's class. So in today's class, I will be starting with the recombinant DNA technology. Now, before going there, as we have studied above in this, that there are two core techniques that are the foundation, that are foundational to all biotechnological processes, and those are either we engineer the genetic material, which is called genetic engineering, and the genetic material is DNA or RNA. So either we play with the genetic material or we play with the everything around it, which means uh, the environment, sterile ambience, tools, techniques, which help us to do this chemical engineering better. Now, I think, uh, did we discuss that bioengineering is just like it's engineering only, but on a biological molecular scale? So, what do engineers do? You must have heard of the term engineering or engineers. So, uh, for example, the most common ones are civil engineer, mechanical engineer, electrical engineer as well to some extent. But there are molecular engineers or chemical engineers. Have you heard of chemical engineers? Do you know what do chemical engineers do? And now there are bioengineers or biotechnological engineers. You can actually do a bachelor's degree in biotechnology engineering. So it's called BTech Biotechnology, which means you basically learn about these things to make a career in it. Now, engineering simply means taking something which is raw and modifying it, its structure, its composition, both. And then with that modification, something new, a new function comes out, correct? So you take bricks and cement, put them together in a way where you support it with metal rods, and you make a structure which is a building. Now, the brick, the cement, and the metal all alone was not serving the purpose, but now the structure that you have created can serve the purpose of being a hospital or a school or a home or anything. Make sense?
Yes.
Yep. Similarly, in genetic engineering, what do we need is we need the genetic material, which is DNA and RNA. Now we are trying to modify them in a way where whatever we need to do, whatever phenotype we need to affect with this gene modification, we do that, then we introduce it or inject it into the host organism, and then we study the change in the phenotype, or we study the new function that the host has now attained. So that's genetic engineering. And for that we need to modify the DNA, correct? So a DNA which is normally present in a cell, let's start with a bacterial cell. So you tell me, where is DNA present in a bacterial cell? I'm taking an example of, let's say, E. coli. So this is the membrane. Where is DNA? Where is DNA in a bacterial cell?
At the end.
No, by where I don't mean the position, because there is no position. It can be anywhere, but where it is. It's the, yeah. Do bacterial cells have nucleus? Bacterial cells are prokaryotic cells, right? So they do not have any membrane bound organelle inside them.
Chromosome?
They have, yes, they have one chromosome. So, or you can say the DNA in a bacterial cell is present, just floating around in the cytoplasm, correct? That region is slightly dense due to the presence of DNA binding and DNA protecting proteins. That's why the region is known as nucleolus, but it does not have a membrane or a defined boundary of itself, correct? But we know that DNA is the genetic material in a bacteria, correct? If I would have asked the same question for a eukaryotic cell, like if I say, where is DNA present in a plant cell, what can you say?
Sorry, sir.
Where is DNA present in a plant or an animal cell, like a human cell or a plant cell, which is a eukaryotic cell?
Say the nucleus.
Just the nucleus?
Oh, yeah, it's also like present in mitochondria. They have their own DNA.
Exactly. And anything else?
Chloroplast.
Chloroplast. So in a eukaryotic cell, if it is a plant cell, DNA can be present in the nucleus. DNA will also be present in the mitochondria. DNA will also be present in the chloroplast, correct? But in a bacterial cell, we know that DNA is just this one single chromosome which is present freely floating in the cytoplasm. But do you know that bacteria also have additional DNA apart from the chromosomes? They have small circular DNA, which is known as plasmid. Have you heard of plasmid? This is the genetic material, which is DNA. This is plasmid. Plasmid is a non-chromosomal DNA. Non-chromosomal circular DNA present in a bacteria. It is an extra DNA present apart from the chromosomes, or apart from the chromosome or the genetic material of the bacteria itself. Have you not studied about plasmid, people, in your junior classes? You must have studied plasmid, right? Yes or no?
So I don't remember.
I don't remember. Okay, no problem. So you can write down some bacteria, not all, but some bacteria have an autonomously replicating, and this is, what do we mean by autonomously replicating? It replicates on its own without depending on, or without independent of the genetic material. Okay, so its replication can take place independent of the replication of the genetic material, the DNA of the genetic DNA of the bacteria. So it is a autonomously replicating non-chromosomal, or you can say extra chromosomal. That's a good word. It's extra. Of course, it is non-chromosomal. It's not, but it is extra, more than the chromosome. Extra chromosomal circular DNA. Now you're going to hear the word plasmid a lot. So just remember this. Is plasmid clear?
Yes, sir.
It is not an organelle. It is not bound by any membrane bound organelle, it is not the DNA of mitochondria or chloroplast or anything like that. It is just naked circular DNA. Now, the fact that it is circular is very important, because if it is circular, so DNA is a double helical structure, right? So which means that if it is circular, it is something, it will look something like this, correct? So what is the benefit of being circular as a DNA? What is the benefit of being circular? If it was just a linear DNA, like a random piece of DNA freely floating in the cell, versus a circular DNA, what is the difference?
It's more stable.
Exactly, it's more stable because it has no loose ends, right? So things can start degrading the DNA from this side or from this side and start reacting to it, correct? And over time it will be gone. You understand?
So
But in a circular DNA, you have no loose ends. It's just like a bangle. It's more stable than a linear form of DNA. So that's where it does not need coating, or it's not present inside any organelle or something, because bacterial cells do not have cell organelles which are membrane bound. It only have ribosomes. Okay. So you will hear the word plasmid over and over again. So just know this. Okay. Now, what we need to do, what we are doing in genetic engineering, is that we are achieving a goal of sending our gene of interest. So let's say there is a gene which I'm interested in. Okay. And this is the DNA fragment on which my gene from this region to this region is a gene X. I'm interested in this gene. Now this gene is present in human, let's say human genome. Now I want to take it out because I want to study this gene, know its effect, etc., what it codes for, protein, etc. So I want to make multiple copies of this gene. This is called gene cloning. Okay, I ask you to go back and read gene cloning, right?
yes,
So the plasmid part is clear, right? Now let's say I want to make multiple copies of this gene X. So what I want to do, what I have to do, I have to take that gene out from where it is and the natural source. Then I have to put it in some other organism or host where it can make it multiple copies. Now the question is, there are two ways to do this. Before we knew plasmid, there was only one way. If you don't know about plasmid, I'm so sorry, then there is only one way. What is that one way? Bacteria divides very fast, right? So if I naturally want to clone a gene, how can you clone a gene? Any idea? You have to make multiple copies of the gene. How can you do that? How a gene can be cloned? We are not making RNA from the DNA. We are making the gene, the DNA copies itself. How will you make multiple copies of the sequence in anything, like in the cell, or how will you do that? Any idea?
Replication.
Perfect, DNA replication. Now, again, but that is just not the answer. How will you do that? If I ask you, if we can somehow incorporate our gene of interest into the bacterial genome. So if I can somehow cut this genome from here, the bacterial genome, it will open, and then put my gene in between and close it. Then bacteria, we know that it divides very fast. Within hours or within days, we will get millions of copies of bacteria, and hence we can get millions of copies of, we will get millions of copies of our gene of interest also, right? Correct? Yes, no?
Yes, sir.
But there is one problem with this technique. What is the problem? What do you think is the problem?
The DNA is circular.
The DNA is circular. Plasmid is circular. And even if like the chromosomal, let's say the chromosomal DNA also is circular. But what is the problem? Why is that a problem? In fact, that's a good thing. It's a stable DNA. The problem is that even though the genome of bacteria is very small, it still have thousands of genes, and we are just putting our one gene inside. Then it will, when it will replicate, it will not be just our gene, it will be the whole genome that's replicating, correct? Because in bacteria, replication occurs when cell division occurs, correct? At the time of the cell division, the cell replicates the DNA and then divide it into two daughter cells. Yes or no?
Yes, sir.
So we will get a million copies of the whole genome, and from there we have to find our gene, isolate it slightly. It's like finding the needle in the haystack and then taking it out. Though there are ways to do that, it can be done. But what would have been a better scenario? The better scenario is right in front of your eyes, marked in blue. What would have been a better
Plasmid.
scenario? Yes. Why is plasmid a better scenario? See, these are the questions for which you have come to the class. If you answer these questions, you will not forget it. If I keep answering every question, you may not remember all of it. So try to participate in the discussion so that you answer the question, so that you remember that I answered this question in Rahul's class, and you will never forget it because it came from your mouth the first. So why is plasmid a better scenario? Because any gene which is in the plasmid, if we have to isolate it after millions of copies are formed, we just have to see it in the plasmid only, like, and plasmid contains very few genes, only the ones that the bacteria has taken up from the surroundings, or the bacteria wants to transfer to some, or some regulatory genes, very few genes, right? So isn't it a better thing? We don't have to care about the whole DNA genome. We can just isolate the plasmid because it will have a smaller size and it's circular. And then if our gene of interest is in the plasmid, it's easier to get that also. Am I right?
Yes, sir.
Yep. But the problem is that this plasmid does not contain the gene X that I want to study. Plasmid has its own genes. So what to do?
Restriction enzyme.
What will you do with restriction enzyme?
Sir, I saw in the net you use restriction enzymes digest some of the DNA of the plasmid, then digest some
Forget
of
about
the.
net. You don't even understand now what is digesting a DNA means, what is restriction enzyme. Biology is as simple as we can communicate. If you start learning biology from the book, if it was that easier, you would not have come to my classes, right? So just answer what you think. If I gave a problem to Atif. See, Atif, I want to take a bag on vacation and I want to take my shoes, but my shoes are not in my bag. So what should I do? What will Atif say?
Search for your shoes and then put them in the bag.
Obviously, search for your shoes, put them in the bag, take it with you. Atif is not going to tell me, okay, I read on the net that there are restriction enzymes and XYZ. It's simple. Gene X is not in the plasmid. Let's put it in the plasmid. First step. Then we'll put the plasmid in the bacteria and allow the bacteria to make multiple copies of the plasmid when it will divide. Yes or no? Was that difficult?
No, sir.
No terminology, no jargon involved. You solved the problem. That's exactly what we are going to do now. So this this whole process of taking a gene of interest, which is a DNA sequence, putting it in a plasmid, which means making making a recombinant DNA, is called recombinant DNA technology. The first thing. Okay, are the words clear to you? Now let's go to the mechanism. So what we do is, any gene that we are interested in. So as I told you that the plasmid is circular. First thing, to put anything in a plasmid, we need to cut both the plasmid and our DNA. So our DNA is somewhere, the gene of interest, this gene X plus our plasmid. Just forgive me for my drawing skills, but you understand this is a circular DNA, right, everyone?
Yes, sir.
Yeah. So this is the plasmid. This is the gene X. I want to put this gene into this plasmid. Now first, this is also somewhere else and this plasmid is closed. So first thing first, to put shoes in the bag, what I have to do is,
Put gene X into the plasmid.
but to put shoes in the bag, I have to open the bag, right? Similarly, I have to open the plasmid, cut open the plasmid. So there's a space. We are not putting it inside here, okay? Don't think like that. We will, because this is also a DNA only, no? It's just a joint circular DNA, like a bangle. We open the plasmid. So I don't know if you can see my fingers. Can you see this shape I have made from my fingers? This?
Yes, sir.
Let's say this is the plasmid. And this pen is the additional gene that I want to clone. I think I should take something smaller, which I can hold. Just one moment. Let me see. Yes. Let's say this cap. This cap is something which I want. This is my gene of interest. This is my plasmid. So first thing I'll do is I'll cut open the plasmid so that I can hold my, I can hold my gene. Can you see this, people?
Yes, sir.
So this is a recombinant DNA. The plasmid DNA carrying our gene of interest. Okay, this is what we have to do. And to do that, now let's come to the word that Atif introduced. And what was the word, Atif? So how will you do that? We use, you said restriction enzymes, right? We use restriction enzymes to cut open DNA, any DNA. Now restriction enzymes, as you know, now that they recognize a specific location on the DNA and that location is recognized on the basis of a sequence. Anything on a DNA is sequence based. So they recognize a specific sequence and then they go and cut at that sequence, because of which the double stranded DNA is cut and open. But tell me, how many cuts do I want to put, I want to make in this plasmid to put my gene X into it? How many cuts will be required? 1, 2, 3, 4, any number of, the less the better, the more the better. What? I just showed it to you using my hand. How many cut did I make to incorporate the gene of interest?
One.
Just one. Yes, just one. So I just want to cut it once. Let's say here, so that it opens. If I cut it at two places, what is the problem? My plasmid is no longer a plasmid. There are two linear chains now, right? So this from here to here will be one chain, and this from here to here will be one linear chain, right?
Yes.
So we want it to stay circular, stay a plasmid. So we just need to create one nick. How many nicks do we want to create to take out the gene X from wherever it is in the genome?
One.
Okay. So if I create one nick, let's say I cut an here, is my gene X free? It is free from this part, but this part is still there, no? So I need to make two cuts, correct? So that only the gene X that I am interested in is free, and now I can put it into this. Is it clear, everyone? These are very simple things. No one pays attention to it, but this is what makes difference in your understanding when you're going to write question answers or understand the process. Has this chapter, it has been covered in your school? Is it done? Donna, Atif, Fatima, is it still continuing in your school, or is it done?
It's for
Biotechnology?
Yes, biotechnology.
No, it's over. We finished portions.
You finished?
The full book. We have just ecosystem left.
Yes, so you must have studied this, right? Did you finish it very early so that you don't remember?
Yes, we finished it pretty quickly.
That's one disadvantage of finishing pretty quickly, right?
So,
You don't even remember what did you finish. So I'm sure that you must have studied, you must have discussed about, the teacher must have discussed about plasmid, restriction enzymes, but all of this seems very alien to everyone because no one is responding. So those, I just urge that those who have, in whose syllabus this is done, please contribute to the discussion, okay, for others, because you will be helping others. Anyone is in whose school syllabus this is still going on or still to get started?
mine. We didn't start this yet.
You have not started it yet. Okay, good.
No.
So for Fatima, I think everyone else should contribute to the discussion so that she can learn. It's the only person in this group for whom this is the first time hearing these words is Fatima. I'll expect that. So I will expect some contribution from others. Though I will take everything from scratch. So my teaching, I will not assume that you know this and I'll skip something, but I'll expect contributions. Okay. Cool. So we first solved the theoretical part of the issue. So theoretically, we solve the problem. We have to cut our gene. We have to put it in plasmid. To cut the gene, we need to cut it at two places, before and after. In between should be our gene. And to cut the plasmid, we just need to cut at any one specific site so that it opens and we can insert our gene in between. This whole process is called making a recombinant DNA. Now, why do we make recombinant DNA? I already told you, for cloning and to clone any gene. Now, what is gene cloning? Just let's define gene cloning quickly. Write down. What is gene cloning or DNA cloning? You can write down. the process of making multiple identical copies. The process of making multiple identical copies of any DNA sequence or a piece of DNA or a gene, anything you can write. I'm having some hot water to help my throat. The process of, what did I say? The process of making multiple identical copies of a sequence of DNA or a gene or a piece of DNA is known as cloning. Simple, right? The only condition is the copy should be identical, no change. Okay, and for this we have to rely on the process of replication, because biologically we know that there is one process which make copies of DNA, and that process is called replication, about which we have already studied in chapter 5. I hope everyone remembers replication. DNA replication, remember?
Yes, sir.
Which enzyme does DNA replication? What is the name of the enzyme which is involved in DNA replication? Whenever someone asks you the name of enzyme, just think about the function process. The process is DNA replication. What is DNA? A polynucleotide. What are we doing? We are polymerizing. It's a polymer. We are polymerizing it, making it again. So the enzyme is called polymerase. And because it is making DNA using a DNA as a template, so it's called DNA dependent DNA polymerase. So DNA dependent DNA polymerase is the name of the enzyme that helps in replication. If the question would have been which enzyme helps in transcription, what would you say? What is transcription? I think all of you are just studying. Yeah.
Copying
Yes, yes.
information from one strand of the DNA into RNA.
Perfect. So we are making a RNA from a DNA. Simple, in simple terms. So what should be the name of the enzyme that facilitates this process?
DNA dependent RNA polymerase.
Perfect. DNA dependent RNA polymerase. If somehow I am making RNA from a RNA, let's say, hypothetically, then it becomes RNA dependent RNA polymerase. And in case of reverse transcription, what is reverse transcription? What is reverse transcription, people?
DNA from
DNA
RNA.
from RNA.
Yes. So what should be the name of the enzyme?
RNA dependent DNA polymerase.
Okay. Was it difficult?
No.
No, right? You just have to think about it, understand the process. Once you understand, your memorizing load decreases a lot. Okay. So you will not be confused in these enzymes from now onwards. Similarly here, we are relying on the process of replication. Now about replication, write down something. What I'm going to tell you next. Has any one of you heard of this term? Origin of replication?
Yes, sir.
Yes. What is it, Atif?
Basically, it's like a place in the gene where replication begins.
Place in the, not in the gene, place in the whole genome, in the DNA, right? In the chromosome. You're correct. So as it says, origin of replication. So let's say this is a DNA thread from the beginning till the end, let's say. So you mean to say that there has to be a place from where the replication begins, right?
Yes, sir.
And in replication, only one strand is, both the strands replicate in semi-conservative method and 2 new DNA strands come up, right?
Yes, sir.
Very good. So where do you think will be that point of origin of replication? Don't you think, like, I think it has to be here. Just start from the beginning and replicate the whole DNA and end here. No? Yes or no? Please do the talking, people. How many origin of replication do you think should be there in one DNA? One long DNA.
One.
One. That's what you logically think. There are many. Okay. There can be origin of replication here. There can be a here. It can be here. Here. There can be many origin of replications in any given DNA. The number varies.
So there's no fixed number.
No, the number varies. That's what I was saying. The number is not fixed. It is fixed for, sometimes it is fixed for particular strains or species, or the cell type or the tissue type where this DNA is. Some cells divide and replicate more. Some cells do not divide and replicate, so they do not have origin of replication in their genome because the cells are terminally differentiated. They don't replicate. So it depends. It varies between cell type to cell type, tissue to tissue, or also organism to organism. But we now understand that whatever is the number, it is very crucial. Origin of replication is nothing but a DNA sequence itself. But that is the DNA sequence. These regions are very important because origin of replication is the point where the whole replication machinery will begin from. Okay, you understand?
Yes,
So these
sir.
are the,
Can
yeah, tell me.
So is there can there be DNA replication like without cell division, or does it only happen during cell division?
DNA replication, it's a good question. It actually happens during this the phase which is a part of the cell division. If you have you remember studying cell division, cell cycle and cell division. Cell division is the very last stage of the cell cycle, correct? First, what happens? The cell grows in size, then it replicates its genome, make everything in the cell double, and then in the last is the cell division. So it happens before that. And there are cases it's possible that you replicate the DNA but do not divide the cell. Do you understand, Atif?
Yes, sir.
So there are three things that are happening: DNA replication, nuclear division, which is called karyokinesis, sorry, and cell division, which is called cytokinesis. So nuclear division, DNA replication happens first, then the nucleus divides into two. So even after nuclear division, the cell may or may not divide. In that case, you will have a multinucleated cells, and we have such cells in our body. Can you anyone tell me where do we have multinucleated cells in our body, where every cell has more than one nucleus?
Centralized cells.
Okay, apart from that, your muscle cells. Don't you think your muscle cells, you have not heard of smooth muscle cells, striated muscle cells, cardiac cells? Anything that needs to generate more power, needs to synthesize more protein, are multinucleated cells. Make sense?
Yes, sir.
The bulk of your body is made up of cells which are multinucleated. So it can, as you said, DNA can replicate without the cell division. Okay, does that answer your question, Atif?
Yes, sir.
but mostly because whenever we study biology, we first study the standard. So DNA replication is followed by nuclear division and then the cell division. Okay, so these origin of replications are the sites, hotspots, which are very important. So write down. In chromosomes, there are specific DNA sequences. In chromosome, there are specific DNA sequences known as origin of replication sequence. Known as origin of replication sequence. It's known as ORI. It's denoted as ORI. ORI. Origin of replication sequence. Which are responsible for the initiation of replication. The initiation of replication. So this part is clear?
Yes.
Yes.
sir.
So now we have one more problem to tackle. Coming back to our solution that we created. Now I added one more complexity to it. For any DNA sequence, so when this gene X was in the genome, in our genome, it was getting replicated because in the genome there was origin of replication somewhere. When I just specifically cut out this gene, it does not have origin of replication now, right? Yes or no?
Yes, sir.
So what to do? What to do? If you don't have something, get it. We also need origin of replication sequence now along with our gene of interest. So we will put a origin of replication in the plasmid along with our gene, or because I told you these plasmids autonomously replicate in the cell. What does it tell you? Plasmid is a DNA. If it is autonomously replicating in a cell, it means that it means what, people? It can mean just only one straightforward thing. Studs are compulsory in a competition, in a tournament to play. And you heard the news that a team is playing the tournament already. What does that mean? Legs are mandatory to walk on your own, and you every day walk on your own. What does that mean? It's as simple. It means you have legs. Correct?
Yes, sir.
Yeah. Very tough question, right? No one was answering. So if plasmid autonomously replicate in the cell, in the bacterial cell, it means that they must be having origin of replication. We just don't know where it is in the plasmid, but it must be having its own origin of replication. Otherwise, it would not have been able to autonomously replicate in the cell. Correct?
Yes, sir.
Yeah. So either we use the plasmid's own ORI, or if we cannot find it, or if we don't know where it is and we want to just use our origin of replication, we can insert our own origin of replication with our gene of interest. Make sense?
Yes, sir.
Okay, cool. So everything set in place. Now let's try doing it. Now let's come back to restriction enzymes. Can I take a 1 minute break here?
Sure,
I'll
sir.
Sure,
just
sir.
I'll just take a couple of minutes off and I'll be back. Okay, just go through the notes. I need some water. Okay, am I audible, everyone?
Mm.
Yep. Okay, everyone is here. I'm back.
Yes, sir.
Yep. Okay. So everything is in the place. Now let's start doing some biotechnological stuff. So it's clear that we want to make a recombinant DNA. Okay, so we need certain tools to do that because we are engineers now, bioengineers. We want to engineer a new DNA using a plasmid. So the plasmid that we are using here, what purpose we are using it for? To put as a vessel, as a bag, right? To put our gene of interest in that and then send it to the host organism, which in this case will be a bacteria, let's say. So it is acting as a vector for us in which we are delivering the gene X of our interest. Clear? So we will often read the word plasmid vector. Okay. Now talking about the tools that we need to do this. First is tools for recombinant DNA technology. So first thing, we need something to cut the plasmid and cut the gene of interest, which we already know is known as restriction enzymes. Correct? Now, what does the word restriction mean? Restriction, to restrict, to cut, right? Now, there are two types. One is called exonucleases, because we are cutting nucleic acid, and it's an enzyme. So ase, the ase word which will be there. So endonuclease, if I remove this S, singular, exonuclease and endonuclease. You can understand the difference. Exo means
Extreme.
exo means outside, towards the outside, right? Outer. And endo means
Inner.
inner. So the difference between an exonuclease and an endonuclease is simple. An endonuclease digests a DNA from outside. So if it is a double-stranded DNA, let's say, it will, this ends, it will start removing the bases from the ends and go towards the inside. Make sense?
Yes, sir.
Whereas what will an endonuclease do is, it will identify some sequence in the center, in the middle, towards the inside of the DNA, and it will come and cut the DNA here. That's endonuclease. Okay, for our purpose we are using endo. We will be focusing on endonucleases, which recognize a particular sequence anywhere in the DNA and can cleave there, can make a cut there. So just to write down the difference. Endonuclease first write exonuclease. Exonuclease remove nucleotides. Exonucleases, EXO, remove nucleotides from the ends of the DNA. From the ends of the DNA. And endonucleases can make a cut endonucleases can make a cut at specific positions. So they also don't cut randomly anywhere in the center, in the middle, at specific positions within the DNA. Not at the ends, but within the DNA. Okay, so that's the difference. Now talking about restriction endonucleases, so we'll be talking about endonucleases, so I'll be calling them restriction endonucleases. So I'll be focusing on two restriction endonucleases here. Because here we are interested in cutting it where our gene is, or in the plasmid, right? So for a plasmid which is circular, you can understand that till it is circular, there is no concept of a exonuclease acting on it, correct? Yes or no?
Yes, sir.
That's why they are more stable. Makes sense why circularity gives more stability to the DNA? Because there is no free end where exonucleases can act. So to cut it, you need an endonuclease. Make sense?
Yes, sir.
Yeah. So the restriction endonucleases. Let's talk about ECO R1. This is the first one. Very famous, very commonly used, and also in your syllabus. Now, and the second one we'll take is HIND2 or HIND2. Now there is a way, there's a logic behind their names actually. So the name ECO R1 is not just a random name. For any restriction endonuclease, when we name it, the first thing here, the ECO, this is telling about the organism, source organism, from where this restriction endonuclease has been derived or extracted from. The second capital letter talks about the strain of that organism, and this Roman 1, 2 or 3 or whatever it is, it tells you the order of what do you call order of isolation, like when it was isolated. So if it is ECO R1, it means that this restriction endonuclease is taken out from Escherichia coli, E. coli. That's why the word ECO, E. coli. R is that strain. In this case, it is the strain RY13. So this R from the strain, and one is it is the first endonuclease which was isolated from this strain of this organism. Is it clear? If you find something written as ECO R2, what does that mean?
The second string.
Not the second strain. It's the second endonuclease derived from the same strain, from the same organism. Okay? It is a second endonuclease, not a second strain. The second strain will be ECO S1 or ECO P1, or if something Am I audible, everyone? Sorry, I lost the network.
Yes, sir.
Am I?
Yes,
Yes.
sir.
Donna, you are the host. Can you make me the co-host, please?
Yes, sir.
Thank you. Yes, now you can see the notebook, everyone, right? Yes, coming back. Sorry for this interruption. Yeah. Now, what I was saying, what was I saying? Yes, if it is something like Eco S or some other strain, then you will say that it is the other strain. Okay. Eco R means E. coli, the strain RY13, this strain. Okay. Similarly, you have one more that you have to study is Hind 2. Now you tell me, what do you think? The first three, Eco, Hin. What does Hin means? Anyone knows the source organism in this case? It's something that you have studied about already. It's Haemophilus influenza. D means the strain, which there is a strain called DS or SD. And 2 means second endonuclease to be isolated. Okay. So there are Hind 1 and Hind 3 as well, but we use Hind 2 mostly. Is it clear, everyone?
Yes, sir.
Yes, sir.
Yeah. Now, there are many such restriction enzymes. Okay, and when I say many, I mean literally. So till now, we have around 1,000 restriction enzymes that we have isolated from around 250 bacterial strains. Okay. Now, what is the benefit of having such a huge library of restriction enzymes? It gives us the freedom to cut the DNA with more flexibility wherever we want, right? You understand? And how these restriction enzymes work, especially restriction endonucleases work, is that they always recognize a specific sequence. Now it can be a 6, mostly it's a 6 base pair sequence. In case of Hen2, it's a 6 base pair sequence. It can be 4 or 8 also for other restriction endonucleases, but let's say it's 6. So that 6 base pair sequence, wherever it is present in the DNA, it will go and cut there. Okay. Now we have to decide according to what gene we want to isolate and put in the plasmid and where we want to cut the plasmid. We select these restriction and endonucleases for ourselves. Okay, out of these around 900 to 1,000 restriction enzymes that we have. So, and in a plasmid, when we make a artificial or when we make a recombinant DNA plasmid vector, we also call it a cloning vector because we are making it. Every component we are fitting in. We are deciding what origin of replication ORI to select that sequence. We will ligate that sequence with the gene of interest. Then we will also select a sequence where we want our restriction enzyme to come and act, or that sequence is already there. And we are designing that whole vector. So it's called a cloning vector. So there are many cloning vectors which are already synthesized and available in commercially. So they're available for research purposes. One of the most famous cloning vectors, most commonly used, which is in E. coli, which is used, is PBR 322. If you go and just put on net PBR 322, you will get a map of a circular cloning vector with many things marked on it. So this is a already synthesized cloning vector in which what it contains it is they give you a origin of replication and they give you space where you want to cut the cloning vector. So it has sequences for almost all common restriction enzymes, which is HIN3, BAM H1, EcoR1, PST1, PVU2, PVU1, SAL1, SAL2. All these sites will be already, all these recognition sites will already be there in that vector. Now, depending on your experiment, what restriction enzyme you are using to cut your gene of interest, the important thing is that you should always remember that you have to use the same restriction enzyme to cut the cloning vector as well. And why is that? Why do you think? Why can't I use different enzymes to cut my gene and different enzyme to cut my vector? That you will get the answer for probably in the next class. But just know that the restriction enzyme has to be same, and depending on that you can use it. Cut your PBR 322 wherever you want, insert your gene of interest, and it's ready for it's ready to be inserted into the bacteria. Okay, now about the restriction endonucleases, one thing that you should remember, they always, each restriction endonuclease only cuts at a specific sequence, and the specialty about these sequences is that these sequences are palindromic sites. You know what is a palindrome, everyone?
Yes.
Palindrome. Yes, what is a palindrome?
It reads the same word of the two opposite sides either way.
Exactly. Can you tell me a word which is a palindrome? Let's say the name Nitin is one which I can think of. It's a palindrome. If you read it in this direction, Nitin. In this direction also it's Nitin, correct? It reads the same in both the direction. So this is a palindrome word. Okay. Now in terms of sequences, so write down restriction endonucleases. Restriction endonucleases. Recognize the DNA sequence. Recognize a palindromic DNA sequence. Restriction endonucleases recognize a palindromic nucleotide sequence or DNA sequence. Now, what is the benefit of recognizing a palindrome? Is that the DNA has two strands, right? One strand reads 5' to 3', and the other reads complementary 3' to 5'. Now let's say here there was a sequence, let's say any sequence, 6 base pair sequence, GAA TTC. Now what will be the complementary sequence? People, what will be the complementary sequence? CTT AAG, correct? Now if you read this 5', this sequence in this direction, it is GAATTC, and if you read this one, this is also the same, right? GAATTC.
Yes, sir.
Yep. So in both the strands, now the benefit of recognizing a palindrome sequence by restriction is the way it cuts is very interesting. It does not make a clean cut like this. It does not cut a DNA like this. This way, cutting a DNA has no use. Like we cannot use it. What it does, on both the strands, it will cut between the same base, between the same two bases. Let's say in this case it is cutting between here A and A. So here also it will cut between A and A, right? But the benefit of cutting between these two is that you can actually do Am I audible? Am I audible, people? Sorry. Yeah. Okay. Cool. Just let me quickly go there. Yes, you all can see the screen. Yeah. Yes. So I was saying that the benefit of cutting like this is that we are left with sticky ends. So in this part, we are left with GA and ATTC. And here we are left with CTT. So GA and CT will be still paired, but this T is free, right? You understand? This
Yes,
T
sir.
here, because of this cut, this is free. And on the other side, sorry, not just the T, but also the A. These two are free because only these two are bound to these two when the DNA is cut. So it will, the DNA will look something like this, smaller, and it has two extra, right? And the other end will look something like this, correct?
Yes, sir.
You understood, people? So these are still ready to unbound nucleotides. This is called sticky ends. So restriction enzymes, when they cut the DNA, they leave sticky ends. Now these sticky ends are very important for us to do this whole process of making recombinant DNA and putting it in plasmid, etc. How, that we will understand in the next class. Okay, so let me just stop here. And if you have any questions, you can ask, people. Do you have any question? Okay, then I'll see you all